the next speaker is Kenneth Hoehn from the Geisel School of Medicine at Dartmouth. And he will talk to us about single-cell B-cell phylogenetics with Immcantation. Kenneth is also a developer of Immcantation and was until recently sitting here in the Kleinstein group.
Thank you, Giselle. So I'm Ken Hoehn. I am an assistant professor in the Department of Biomedical Data Science at Dartmouth. And today I'm going to talk about single B-cell phylogenetics with Immcantation. So the reason we can use phylogenetics and molecular evolutionary techniques to study B cells is because B cell diversity is shaped through mutation and selection. So both rapid somatic hypermutation and antigen-driven selection work together to form what are called B cell clones, which are cells that relate to each other by point mutations from a common naive B cell rearrangement. And the goal of B cell phylogenetics is to reconstruct the series of mutations leading from the naive B cell to the sequences that we observe, and to use those trees to understand how the underlying biology is being altered. Now, one of the most interesting or potentially useful things about single-cell sequencing is that it can provide paired heavy and light chain BCR sequences. So historically, only heavy chain sequences were used to build lineage trees, or at least primarily heavy chain sequences were used to build lineage trees. And this is because bulk sequencing separates heavy chains and light chains from the same cell. And because of this, we're not able to determine which heavy chain belongs to which light chain. With single-cell sequencing, we can now obtain paired heavy and light chain sequences from the same cells. And some studies have incorporated both of these chains by concatenating the sequences and treating them as part of the same gene. However, it's unclear how to best build B-cell lineage trees with this paired data. And I should mention that most of this work was actually done by Cole Jensen, who is currently a PhD student in the Kleinstein lab. So one reason we might want to have more complex models for incorporating light chain sequence information is that light chain BCR sequences have distinct mutation patterns from heavy chain BCR sequences. So it's been known that light chains have a lower rate of mutations compared to the heavy chain. And to confirm this, we looked at two empirical data sets of COVID-19 patients and an influenza patient, and quantified the level of somatic hypermutation in the heavy chains for each clone and the light chains for each clone. So each of these dots represents a clone, and we can see that there is a relationship between the level of somatic hypermutation on the heavy chain and the level on the light chain. However, the light chain is much less mutated, about 50% the mutation level of the heavy chain. And the results are consistent in this larger influenza data set. It's also been known that light chains have distinct somatic hypermutation hotspots and cold spots from heavy chains. So it's important that simulations and methods take these biological differences into account. Now, to benchmark phylogenetic methods, we really need some kind of simulation framework. And currently, there was no existing software for simulating paired heavy and light chain BCR evolution. So we used a combined approach, including BCR Phylo, which was developed by the Matson group, and the Shazam package, which has functions for simulating sequences. So with BCR Phylo, we generated tree topologies and branch lengths. We then used the shmulate tree function in Shazam to shmulate heavy chain sequences by iteratively adding mutations down each of the trees. We also simulated light chain evolution, but in order to do that, we shrunk branch lengths by 50% and then simulated down. This meant that the light chains that we simulated were about half as mutated as the heavy chains. We also used previously published heavy and light chain hotspot models, which are available in Shazam. And this gave us simulated data that looked a lot like our empirical data, where our heavy chains are about twice as mutated as our light chains, and we have a similar level of somatic hypermutation as in our empirical data sets. And we can use this simulated data to test whether including light chains improves tree accuracy. We looked at multiple different phylogenetic methods, all of which are supported by the R package Dowser. And rather than going into all of these, they separate into three main frameworks. The first is maximum parsimony. And in this, you find the tree with the fewest mutations. It's a very intuitive way of building trees, and a lot of B-cell phylogenetics packages use this means of building trees. We also tested methods that are single partition maximum likelihood. So in here, we use a nucleotide model of nucleotide evolution and find the tree that maximizes the likelihood of the data. Importantly, these approaches, which are single partition and are generally the standard maximum likelihood approaches, treat heavy and light chains as if they're part of the same gene or partition. And finally, we have multi-partition maximum likelihood methods. So these methods separate the heavy and the light chains on separate gene partitions and allow the branch lengths of the trees to be different between the two. So these methods are able to estimate separate branch lengths for the heavy chains and the light chains. Importantly, we don't estimate a separate branch length for each branch for the light chain. We instead multiply the light chain tree branches by a single scaling factor. So the light chain branches are shrunk proportionally relative to the heavy chains. And this allows us to test heavy and light chain built tree building across multiple phylogenetic frameworks. Now, one of the metrics that we used to quantify the difference in tree building accuracy is called the Robinson fold distance. And this measures the difference in tree topology from the true tree. So the Robinson fold distance is basically the edit distance from two trees, in this case, from the true tree to the tree that we build. And it's the number of rearrangements you have to make in the tree in order to transform it into the other one. So a lower Robinson fold distance indicates a more accurate tree topology. Importantly, this metric doesn't include branch lengths, but it's still a useful way of summarizing tree accuracy. And we can see in these plots that trees built using only the heavy chains have a much higher estimated Robinson fold cluster distance from the true tree compared to trees built used with both the heavy chain and the light chain. So in this method, Van-Goren parsimony, we can see that on almost every clone, adding the light chain gives you a more accurate tree topology. And this is the same in both maximum likelihood and multi-partition maximum likelihood. And in all of the methods that we tested, on average, you get a significant improvement in tree accuracy when you include both chains. Importantly, all of the methods we tested using both the heavy chain and the light chain were more accurate than all of the methods we tested using only the heavy chain. So using the paired chain significantly improves tree accuracy in the methods we tested, at least in this simulated data. Now, in that simulated data, every heavy chain was paired with a light chain. However, in real data sets, we often have missing light chains. This could be due to dropout in single cell data, but also from combining bulk and single cell data. In our framework, missing chains are included in the alignment as ambiguous in nucleotides. So here is our fully paired multiple sequence alignment, where every heavy chain has a light chain. And here is the multiple sequence alignment where some light chains are missing. So here we have our single cell data, and we have our bulk data, which only has heavy chains represented. Now we can use our same simulations to investigate how different methods perform when some light chains are missing. And we found that when light chains are missing, things get a little bit more different between the methods. In the simulation, we discarded different proportions of light chains, and we evaluated the RF distance for topological accuracy and the tree length, which is the sum of branch lengths. Here I've shown the Robinson fold distance. So the y-axis is the mean RF distance in the simulation run from the different methods. And the x-axis shows the percentage of light chains that are removed from the simulation. So at this data point, we have our fully paired data, and that's the same as the previous figure that I showed. And over here, we have our heavy chain only data. And at this data point at 95, for those simulations, 95% of the paired heavy and light chains had their light chain removed. So in this 95 data point, almost every cell had a missing light chain. And we can see that as more light chains go missing, the topological accuracy of the trees decreases. But we still get an improvement in tree accuracy up to about 30% missing light chains. So we can conclude that topological accuracy can still be improved even if some light chains are missing. And that this approach is relatively consistent among methods, though some seem to perform worse when about 50% of light chains are missing. When we look at tree length, the methods separate quite dramatically. So tree length is the sum of all branches within a tree, and it represents the total number of mutations that happened within a lineage. So here on the y-axis, we have the proportional error in tree length estimation, which is the difference between the true tree length and the estimated tree length. And again, on the x-axis, we have the percentage of light chains that are missing in the simulations. So we can see that when we build trees using maximum parsimony, we almost always underestimate the tree length, especially as more light chains go missing. When we use single partition maximum likelihood methods, we get the exact opposite problem, where we overestimate the total tree length. So these trees have more mutations than they really should. But when we use multi-partition maximum likelihood methods, we're able to accurately estimate the tree length across all of these different proportions of missing light chains. And these results are explainable based on how these methods deal with missing data. So maximum parsimony is always just trying to minimize the number of mutations. So if there's no sequence, there's no mutations, and it predictably underestimates the total branch length. In single partition maximum likelihood, it assumes that sites are missing randomly across both chains because it considers them to be part of the same gene. And because of this, it effectively imputes mutations into the missing light chains at a similar rate as the heavy chains, and that causes it to overestimate the branch lengths. In multi-partition maximum likelihood methods, however, we're able to account for the fact that the light chain evolves more slowly and more accurately estimate the branch length for those missing light chains. So among these methods, only multi-partition models are able to accurately estimate branch lengths when some light chains are missing. So that is the results that we found in incorporating light chains into phylogenetics and looking at different tree building methods. But I also wanted to talk about different aspects of the Immcantation pipeline that are important to keep in mind when doing B-cell phylogenetic analysis, and also how these have been changed in recent releases to incorporate this data. So the first step in B-cell analysis is pre-processing, which is read assembly and quality control. And this is normally done with Cell Ranger, Presto, or Trust4 if you're using RNA-seq data. And this is really important because bad sequences will give you bad trees. So it's really important to do good read assembly and good quality control. Next is VDJ alignment, where we match to germline databases, either the IMGT or OgreDB. And this is done with IgBlast or IMGT VQuest. And this is important because it gives us the germline alleles and IMGT-gapped BCRs. And related to this, we can also detect novel alleles and genotypes for individuals through tools like Tigger, Piglet, and Rabbit. Next, we have to combine the data. So this is to merge the data from different sources, either from different time points and tissues, but also cell type annotations. So annotating BCRs using cell type annotations from RNA-seq. And we don't have specific functions for that, but it's easy to do that using base R functions like match. And this gives us annotated BCRs and allows us to draw clones between samples within an individual. And next, it's important to infer clones. And this is important because it separates B cells into clones which descend from a common VDJ rearrangement. And this process uses both the heavy chains and the light chains. And it's important to allow connections within individuals, but usually not between individuals. And the packages used for this are scoper and sometimes defined clones if you're using a very big data set. So this is very important for phylogenetics because trees are really built within clones. And within Dowser itself, we've added features for including light chains for B-cell tree building. The first is in resolving light chains. So some cells have more than one light chain. And in order to include them in tree building, we need to ensure that each cell has at most one light chain and that those light chains descend from a common VDJ rearrangement. So this function ensures that each cell has one heavy chain and at most one light chain. Next, we reconstruct the clonal germlines. So this is done for both the heavy chains and the light chains and use the create germlines function in Dowser. And this is important because the germline sequence is used to root the tree. Next, we build the trees, and it's kind of obvious why that's important for phylogenetics. And Dowser supports multiple different tree building methodologies like maximum parsimony, maximum likelihood in both single and multi-partition. And we then are able to plot the trees and analyze things like evolution over time, migration and differentiation, and other things by visualizing this information at the tips and in using statistical techniques, which we've also added to Dowser. So just to summarize, phylogenetic trees use mutations to link B cells to a common ancestor. We can now build them with paired heavy and light chains. All methods are improved from using these chains, but some give highly biased branch lengths if some are missing light chains. And Immcantation supports start-to-finish single B-cell phylogenetic analysis with both chains. And if you're interested in reading further, all of these results and more are detailed in our recent preprint for inferring B-cell phylogenies from paired heavy and light chain BCR sequences. If you're interested in B-cell phylogenetics with single-cell data in general, Steve and I recently published a review paper in Trends in Immunology on how single-cell data can be used to improve B-cell phylogenetics and generally how this process works. And we also have some methods that have been published recently on using B-cell lineage trees from different tissues and cell types, as well as incorporating data from different time points. So with that, thank you for listening. These are links to the software that I've talked about. This is my contact information and lab website. We're currently recruiting postdocs and programmers, so if you're interested in doing this kind of work, get in touch. This is where I am broadcasting from. As you can see, we have a lot of trees. And I want to give a big thanks to Cole Jensen and Steve Kleinstein and all of the other Kleinstein lab members who've helped in making the new releases of Immcantation single cell compatible. With that, I can take questions.
Thanks a lot, Ken. There's a couple of questions in the chat. The first one of them relates to the S chain frequency in the light chains. And the question is whether the frequency is lower in the light chains due to a general lower frequency of hotspot motifs there, or is it because a lower overall S chain rate there?
Yeah, I'm not sure. It's certainly a very consistent pattern that we've observed, but I think the explanation for it is elusive. So I'm not entirely sure why that is.
Okay, thank you very much again for the talk. There's another question, but due to the time reasons, we would ask you to please answer it directly in the chat.